Okay, good morning everyone and welcome to this morning's keynote lecture. My name's Clive Page and it's my pleasure this morning to introduce our keynote speaker, Professor Clifford Woolf. Clifford was born and educated in South Africa, has trained in medicine and has a PhD in neurobiology, and for a number of years was the professor of neurobiology at University College in London. He then became the Richard J. Kitts Professor of Anesthesiology Research at Massachusetts General Hospital and Harvard Medical School in the US, and he's currently the F.M. Kirby Neurobiology Center Director at Boston Children's Hospital and a Professor of Neurology and Neurobiology at Harvard Medical School. Dr. Woolf's many honors and prizes include membership of the American Academy of Arts and Sciences. He's a Doctoris Honoris Causa from the University of Lausanne in Switzerland, a Jill Distinguished Scientist Award, and he won the Reeve Irvine Medal. He works on adaptive and maladaptive plasticity in the nervous system, and today we're very pleased to have you, Clifford, to talk about cell-selective screens for novel analgesics. So welcome, and we look forward to hearing from you.
Good morning. Thank you very much. It's a pleasure to be here. What I'd like to share with you is a platform that we've put together to discover novel analgesics without an abuse liability. When I was invited to give this presentation, the focus was on cell-selective approaches to screening for analgesics, but the platform, as you'll see, has expanded somewhat. Just to indicate some of my potential conflicts, the only one that is relevant to this particular presentation is actually a company called Black Box Bio, which is developing some of the technology that I'll indicate for measuring what we call the body language of pain in rodents. So pain really, for many years, was seen to be a uniform experience, but we now appreciate that there are multiple different kinds of pain with different mechanisms. And nociceptive pain is the physiological pain that warns us of potential danger in the environment. Inflammatory pain is that pain associated with immune responses to tissue injury or pathogen invasion. Neuropathic pain is that pain associated with damage to the nervous system, both the peripheral and the central nervous system. And dysfunctional pain is a complex disorder resulting from the malfunction of those nociceptive circuits in the central nervous system. Pain is an enormous burden. A review in Nature some years back actually indicated that the cost of managing pain is much higher than heart disease, cancer, and diabetes, which is quite extraordinary. And one of the major problems associated with pain is that prescription opioids have contributed to the opioid crisis, which is particularly a problem in North America, but throughout the world. And the data indicated on the right shows the number of individuals who die from opioid abuse. And in consequence, I'm highly motivated to try and identify drugs that can eliminate the need for us ever to use prescription opioids. In addition, we have a real problem, which is that the efficacy of existing drugs is really rather poor. This is a meta-analysis looking at the long-term pain control of patients with knee osteoarthritis, and the table indicates all the different interventions that have been used, and essentially, the line in the middle shows whether there is an improvement in the patient's pain condition, and essentially not a single one of the treatments in terms of chronic management of knee OA is effective, and that is pretty much true for most pain conditions. So we have an enormous need, and in consequence, from a pharmacological point of view, an enormous opportunity to try and develop novel analgesics. So the challenges in the pain field are numerous. One of them is, how do we diagnose pain? What kind of pain do our patients have? How do we measure it? Pain is a subjective experience, which makes it very difficult. We rely on our patients to report their pain, but how do we measure pain in a newborn infant who cannot report their pain? And it is highly variable. So that is a separate matter, but one that and requires novel approaches. What I'm going to focus on is the challenges of treating pain, and in particular, what are the opportunities for developing effective and safe analgesics, and as I've indicated, with a strong motivation to eliminate the need for prescription opioids. And that's what I'm going to focus on here. The traditional approach to drug discovery across all disease states has been to identify targets, develop assays, to then identify compounds that selectively act uniquely on those targets, and then to perform preclinical efficacy and safety studies. And that is the standard approach. Frankly, this has not worked in the development of analgesics. If you look at how many novel analgesics have been developed in the last 3 decades, there are a tiny handful, and even those are not very efficacious and have quite serious adverse effects. So I think it's time for us to contemplate an alternative strategy. And illustrating the problems with this, there have been major efforts to identify particular targets that may play a major role in pain, and in particular the ion channels that may contribute to different pain states. One of the major focuses has been on the voltage-gated sodium channel Nav1.7, which is uniquely expressed in those sensory neurons, the nociceptors, that are activated by noxious stimuli and trigger pain. And there are human mutations in Nav1.7. A loss of function mutation in this channel results in congenital insensitivity to pain, whereas gain of function mutations produce a number of different syndromes like primary erythromelalgia, where the patients have spontaneous severe pain. And this the industry found very exciting, and as a consequence devoted enormous effort to the development of selective Nav1.7 blockers. And in particular, Pfizer has spent an enormous effort on the development of Nav1.7 blockers, and frankly, they have failed in the clinic. So the single target approach has not been very effective. There is another voltage-gated channel, Nav1.8, which a company, Vertex, which is also based in Boston, where I am, has some hints of promising results, but again, the efficacy signal is rather limited. So our strategy has been to say, well, let's not restrict ourselves to a single target. We certainly want to identify nociceptor selective targets, and to do that, we have used a differential expression of transcripts that are expressed selectively in those sensory neurons that initiate pain, the nociceptors, versus other excitable cells, such as in the spinal cord, in the heart, etc. And we have identified multiple potential targets that are present there. But instead of going for drugs that act only on single targets, what we are trying to do now is identify compounds that act on multiple targets, and in this way may improve the efficacy and hopefully will also reduce the adverse effect profile. So what I'd like to share with you is the platform that we've put together to do this. And we've developed a series of toolboxes to do this. One of them has been to develop a means of looking at ion channel modulators using thallium flux assays. And I'll show you in a moment the way we can do this, but this enables us to screen hundreds of thousands, if not millions of compounds to see if they can act on the multiple targets that we have identified as being nociceptor selective. We've also devoted considerable effort to phenotypic screening because we feel that target-based screening is inadequate by itself. And one of the advantages now is that we can now generate human nociceptors from stem cells and use them in assays, and can compare the activity of these nociceptors versus other excitable cells such as motor neurons, cortical neurons, and cardiomyocytes. And we can record the activity in many different ways, including multi-electrode arrays, automatic patch clamp, and this is enabling us to take a fairly sophisticated approach to the identification of compounds that can suppress activity in nociceptors. And then the big challenge is how do we then determine whether the hits that we get have efficacy? And this has been a real problem because how do you measure pain preclinically in rodents, for example? As I've indicated, pain is a subjective experience. You cannot ask a mouse or a rat, how much pain do you have? And therefore, the approaches have been very crude. They've required the application of noxious stimuli and looking at withdrawal reflexes, the same as we would experience if you had a pinprick or touched something that was too hot or too cold. But frankly, that is not the clinical pain syndromes. And therefore, we have devoted considerable effort to using machine learning approaches to measure changes in the behavior of the animals that reflect what the animal is feeling. And I'll share that with you. So our ion channel high throughput screening has adapted this thallium flux assay. This was originally developed for looking at potassium channel activity, but we have managed to adapt it for both potassium, calcium, and sodium channels. And the beauty of this is that we can run screens in 384-well format, so we can literally run many thousands of compounds on a day factor, and the signal is extremely good, as indicated by the Z prime factor. I must admit, as when I was at University College London for many years, I'd never heard of a Z prime, never thought of it, but now I'm totally obsessed about it. Z prime is the absolute clear signal that indicates that your assay, there's no noise, and that the signal that you're getting represents either the activation or blockade of particular channels. In addition, we now have moved away from manual patching of single cells to automatic patch clamp data. My colleague Bruce Bean at Harvard Medical School has been running this, and we are now able, again, to use automatic patch in 384-well format, and can therefore take the compounds that we identify from our thallium flux and test exactly what they do on individual channels that are heterologously expressed, as well as on the human neurons as well. So what I'd like to share with you, though, is how we can measure pain preclinically. And there are two approaches to this. One is in vitro, and the other is in vivo. And for in vitro, we feel the best approach is using human nociceptor assays. And we've been working in collaboration with the translational arm of the NIH called NCATS to develop new differentiation protocols that enable us to make human nociceptors, and from that can develop assays that enable us to see whether the hits that we get from our thallium flux or automatic patch clamp do suppress the activity of human nociceptors. This differentiation protocol has just been published very recently for anyone who is interested. However, when we run our screens, we don't see this just as a primary screen looking for compounds that suppress the activity of nociceptors. We feel it is as important to run counter screens on other excitable cells, such as cortical and motor neurons and cardiomyocytes. So we run the compounds on all 4 cells simultaneously, and if we find a compound that suppresses the activity in all 4, then it's eliminated. We are looking for compounds that uniquely and exclusively suppress activity only in nociceptors. So this represents a phenotypic screen, and as I'll indicate in a moment, it enables us both to test compounds that we can identify from target-based screens, but also we can directly run screens on novel compounds on these cells without targets, so a non-target-based approach to drug discovery, which the pharmaceutical industry, frankly, is not very happy about. So how do we make these human nociceptors? Well, we start with induced pluripotent stem cells. We then make precursors, which we call nocispheres, which go through a stage of neural crest-like development. And finally, we can differentiate them into nociceptors that express all the transcripts that we know are present in human nociceptors. Our collaboration with NCATS has enabled us to have access to a fully automated approach to the generation of these cells, and this enables us to make literally millions of these cells, so we can really run very large screens in a very reliable fashion. There's minimal batch-to-batch variation in the properties of these cells. This illustrates the differentiation protocols, starting from the human pluripotent stem cells through the nocispheres, which are the early developmental stage, leading to the neural crest-like stem cells, and finally the nociceptors. And for those who are interested, our paper just published a few months back has the full details, and if anyone's interested, I'd be happy to share with you how we do this differentiation protocol. But as illustrated, after several weeks, we have a pure set of neurons that express the key markers that are present in nociceptors and have the functional properties of nociceptors. So we now have the means to develop assays to identify compounds that selectively act only on these neurons. We can monitor, for example, the expression of markers of sensory neurons, such as brain 3A, which starts off relatively low and then reaches a point as the cells mature, whereas the SOX10, which is a neural crest marker, starts off high and drops off as the cells mature. We can look at the expression profile of the cells at the different stages. So we start off from the pluripotent stem cells. We see high expression of pluripotent gene markers, as indicated at the top. And then there is the expression of neural ectoderm markers, then neural crest, and then finally at about 12 to 14 days, we start to see the expression of nociceptor transcripts. Excuse me. This particular differentiation protocol does not make a single type of nociceptor. It makes all the major kinds of nociceptors, the peptidergic and non-peptidergic, as well as some of the non-nociceptor sensory neurons as well. What can we use these cells for? Well, as I've indicated, we are using them for phenotypic drug screens, and this enables us to conduct two kinds of drug screens, one of which, as the title of this talk indicates, are screens that are cell-selective. We're looking for compounds that act only on one cell type and not on any other. And as I say, that can be target-based. You can do preliminary studies identifying targets and looking for compounds that act on them, or it could be independent of targets. You're just looking for any compound that has a profile of activity such that it reduces the activity in this cell but has no effect on any other cell. The other kind of screen one can do is disease phenotypic screens that, for example, there are, if one looks at type 2 diabetes, about half of those patients with type 2 diabetes develop peripheral neuropathy, and half of those who develop peripheral neuropathy develop neuropathic pain. if we can identify a phenotype, a hyperexcitability phenotype that reflects the susceptibility to that, we can then also run screens to see if we can reduce that risk of the development of diabetic neuropathic pain. So at the same time I was invited to give this talk, we wrote a review in Science Translational Medicine that looked forward to the approaches that we could adapt, and essentially this summarizes our approach, that we start from somatic cells from a patient, we then convert them into pluripotent stem cells, and from that we can then differentiate them into multiple different kinds of cells, as I've indicated. This then enables us to model disease. Potentially one can consider cell replacement therapies and gene therapies, as well as identification of new drug targets. How about the disease modeling? Well, here's an example. I indicated before that NaV1.7 was a very popular target for industry based on the fact that gain-of-function mutations produce spontaneous pain in patients. Can one capture that, this in these patients? And here is multi-electrode array recordings. We've grown the cells on top of a multi-electrode array device, and we can record the activity of nociceptors without this mutation. And you can see there's very low activity because nociceptors require a noxious stimulus to be activated, so they have minimal spontaneous activity. Whereas if we introduce the gain-of-function mutations, you can see by the color reflecting the spontaneous firing of these cells, that these cells are firing spontaneously. So we have captured in a dish some of the key features that are present in our patients as well. We can also model inflammatory pain conditions. A feature of inflammatory pain is the reduction in the threshold required to produce pain. All of us who've had injuries or an infection are familiar with the fact that instead of a noxious stimulus being required to produce the pain, now stimuli that would normally be innocuous will produce the pain. Touch of the skin, or after a sunburn, mild warm stimuli produce pain. And this is evoked by the presence of inflammatory mediators produced by the immune cells, which act on the nociceptors and sensitize them, reducing their threshold and increasing their excitability. And we can model this by looking at these same human nociceptors and add inflammatory mediators to the culture medium, and when we do that, their excitability increases. So again, we can model inflammatory pain conditions. What about using this approach for drug screening? Well, there are several different ways we can do this. One is a phenotypic screening for targets, and the way one can do that is to use libraries that are annotated for activity on particular targets. There are now several thousand compounds that are available that are known to act on different targets. Many of these compounds act on multiple targets, but nevertheless, you can run a screen on the human nociceptors looking for which one of these annotated compounds suppresses the activity, and from that you can work out whether target A may be involved in generating the excitability of these neurons. And then once you identify that, you can then validate it by knockdown or other approaches. The other approach is actually using these cells for phenotypic screening, starting off with chemically diverse compound libraries where you don't know which target these compounds may or may not act on, but run the screen against these human nociceptors, looking for hits that suppress the activity selectively of these nociceptors. And this is certainly what we're using in our platform. And then once you find a compound that has this profile of activity whereby it suppresses activity in nociceptors with minimal or no activity in other excitable cells, one can then use medchem optimization to improve the drug-like properties of the cell and hopefully the potency of the compound as well. Again, this is something the pharmaceutical industry is not keen because they like structure-activity relationships to do their medchem, but we've been working with professional medicinal chemists now who are very happy using these phenotypic screens where you don't know what the target is, but you know exactly the profile of activity that you want. And then one can also use these cells as counter screens. If we run our thallium flux or automatic patch clamp and we get promising hits, we can then confirm the activity of these hits on these human neurons. How do we run these screens? There are a number of different approaches. One of the easiest ones is to use GCamp as a calcium reporter, which is a calcium reporter. When a cell is depolarized, that activates voltage-gated calcium channels, causing an influx of calcium into the cells. That increase in calcium can then be detected by GCamp as a fluorescent signal. And this can be run, again, on 384 well format. And as I've indicated, we run that on nociceptors with counter screens on other excitable cells, because our goal here is to look for compounds with a selective activity only on nociceptors. And as illustrated here, you can see the kinds of signals that we see. And when we give tetrodotoxin, which is a non-selective sodium channel blocker, it eliminates the activity of these neurons. But we are looking for compounds that will eliminate the activity exclusively of nociceptors and have no effect on any other excitable cell. We have been running these screens, and I apologize, this is work that is not yet published, so I can't identify the compounds yet, but just to illustrate that we are identifying compounds that have exactly the desired profile we're looking for. They suppress the activity of nociceptors without any activity or minimal activity on cortical and motor neurons. And we can confirm this with a patch clamp and by MEA as well. And here is an illustration of one of these hits where we've taken the compound and we're testing its activity on these human nociceptors using multi-electrode array recordings. And you can see that when we start with our DMSO controls, we have activity pre and post DMSO application. We then give the compound at a series of different concentrations, and you can see that at 10 micromolar of this particular compound, there's complete elimination of the activity of the nociceptors, and this particular compound has no activity on cortical and motor neurons. So human nociceptors can be used in multiple ways, can be used for this screening, as I've indicated, the phenotypic screening. We can also use them to model disease and try and begin to address the question, what drives pain? We can also identify patient susceptibility, and we are running screens looking at chemotherapy-induced peripheral neuropathy. It turns out that something like 40% of patients who get chemotherapy for cancer develop peripheral neuropathy, and in about 10% of the patients, the neuropathy is so severe that they terminate their treatment. And can you imagine that as a patient trying to have your cancer treated, but stopping the treatment because the peripheral neuropathy is so severe? There is no, at the moment, there's no way of identifying who's at risk of the development of CIPN, nor is there any treatment to prevent that. And we are now running screens on our human nociceptors using chemotherapeutic agents such as paclitaxel, which reduce the growth of these neurons, and we are looking for neuroprotectants that can prevent this chemotherapy-induced peripheral neuropathy. I've indicated diabetic neuropathy as well, and we are beginning a study with Dave Bennett at Oxford, whereby we are looking at patients, at iPSC lines from patients who have diabetic neuropathy with and without pain, and are identifying phenotypes that reflect the susceptibility, and then can run screens to see if we can reduce the risk of the development of this neuropathy. In the future, we also anticipate that we will be able to personalize treatment, whereby we may take a patient, take their white blood cells, turn them into pluripotent stem cells, differentiate them into human nociceptors, and then identify which set of existing drugs act most effectively on that patient's cells to reduce that activity. So we can identify the responders to particular forms of treatment using this approach. So in addition to this in vitro approach to measuring pain preclinically, the challenge, as I've indicated, is what about once you find a promising compound from this phenotypic screen assay, how can you potentially identify whether it's going to actually work in vivo? And what I'd like to share with you is this machine learning approach to capture what we are calling the body language of pain, which really is those sets of behavioral changes that we anticipate reflect the presence of pain, that when a mouse or a rat is in pain, it will change its behavior in very subtle ways, which machine learning enables us to detect. How can we measure this in a mouse? And I'd like to share with you a strategy that we've developed to do this. And for those of you who are interested, we've published this in December last year, this automatic preclinical detection approach to pain in mice. This enables us, as I share with you, the ability to identify ongoing and spontaneous pain. I mentioned earlier that the standard approach to measure pain preclinically is to apply a noxious stimulus and look for a reflex response, a withdrawal response. But most patients have spontaneous pain, and there are no measures of spontaneous pain. Yet, I'll show you that with our novel approach, we can pick up spontaneous pain in our preclinical models. We can also model clinically relevant pain conditions rather than just an application of noxious stimuli. We can look at inflammatory pain and neuropathic pain and other forms of pain as well, such as joint pain and in due course, hopefully, chronic low back pain. The advantage of this is we don't require an observer. This body language of pain is detected automatically using machine vision, and that is important because mice are fearful of predators, and they see us as potential predators. So if you measure behavior in a mouse, your presence is going to make the mouse extremely stressful. In addition, almost all the measurements are done in the light, except mice are nocturnal animals, so you have again, increasing the stress of the mouse, whereas the approach that I'll share with you enables us to measure the behavior without an observer in the dark. And in addition, the technology has now been developed such that we can do large-scale analgesic drug screening. So this is the technology that was developed. We call it the palm reader, and this was developed by a graduate student in my lab who actually happens to be in the audience here, David Roberson. And we called it the palm reader device, and it has two aspects to it. One is to measure the spontaneous behavior of the animal, what we call the body position or the body pose. But in addition, David introduced the possibility of measuring surface contact as well by using frustrated total internal reflection. So we illuminate this box with near-infrared light, which the mouse cannot see, so it's completely dark to the mouse, but we can see the body position. And if we apply this near-infrared light to the side of the glass surface, wherever there's contact with the surface, you can see a luminescent signal that reflects surface contact. And this enables us to measure mechanical pain hypersensitively. We're all familiar that if you have pain, you are hypersensitive, and we can now measure that hypersensitivity to contact in an automatic fashion. And this is what it looks like. The EFTA signal, the surface contact, is illustrated here in purple. You can see all the pores as the animal is exploring. The animal is not restrained, it's freely moving, it's doing whatever the mouse chooses to do, and hopefully that will include changes in its behavior if it is in a disease state. We can measure exactly the changes in body position, and there are a number of strategies, machine learning algorithms to do that, including deep lab cut, which enable us to measure exactly the relative movement and exactly what the animal is doing over time. The surface contact, the pore luminescence signal, reflects exactly the force applied by the mouse on the surface. As illustrated over there, you can see there's the pore luminescence signal is linearly related to the force applied to the surface. And as illustrated over here, you can see the change in pore luminescence signal of the two hind paws, and as a healthy, normal animal is walking, rearing, pausing, and you can pick up all these changes in its surface contact automatically. So to illustrate the potential value of this approach, we've tested changes in what we call the pore luminescent ratio, whereby we apply inflammation to one of the pores and leave the other one intact, and we can then look at the ratio between the pore luminescence signal of the two pores, the inflamed pore versus the non-inflamed pore. We produce inflammation by intraplantar injection of zynosan, which is a component of some pathogens, and then we are looking for the effect of non-steroidal anti-inflammatory drugs, COX-2 inhibitors. In this case, we're using ketorolac to see if they can reduce the effect of the inflammation on surface contact. One of the problems in preclinical studies has been that the exposure required to produce analgesic signals is typically 10 or even 100 times higher than the exposure patients have. For example, the standard dose of morphine given to a mouse is 5 milligrams per kilogram. If you gave that to a human, they would stop breathing and die immediately, and yet that is the analgesic dose in mice. The standard dose of non-steroidal anti-inflammatory drugs that are given to mice to see a signal, again, are 20 times higher than that in patients. Is that because the animals require higher exposure levels, or is it because our measurements are too insensitive? Well, we set out to explore that, looking at our pore luminescent ratio. So we have a baseline recording where the pore luminescent signal from the two hind pores is equal in the control state. They walk and they expose each pore to the surface in exactly the same way. We then give zynosan in one pore, causes inflammation. The animal then avoids surface contact of that inflamed pore, and consequence, the pore luminescent signal is reduced by half. We can then apply our drugs at different dose levels, and you can see that we have applied ketorolac here at 3 doses, 1, 3, and 10 milligrams per kilogram, and saline. These are all done completely blinded. And all 3 doses produce a significant return of the pore luminescent ratio back to the control states. When we look at the standard measure, which is applying a von Frey filament, which is a nylon filament that applies a force to the surface, we see no significant, there's a hint of some change, but no, it is not significant. So our standard measures cannot pick up the analgesic effect of ketorolac. However, this pore luminescent ratio can pick it up. What about picking up other features, such as sedation versus analgesia? And this is a real problem, because many of the compounds that are tested may be producing a change in the behavior, not because they're producing analgesia, but because they are changing, for example, the state of alertness of the animal. And to illustrate that here, we'll show the effects of the application of diazepam to mice in the same intraplantar zynosan model. And as illustrated here, we're measuring the distance traveled. We're the mouse is running around in the chamber. We then give Zynosan. That reduces the amount of travel the mouse has. Its paw is painful, so it runs around less. And then we give diazepam, and the animal stops moving almost completely. However, when we then look at the paw luminescent ratio, the Zynosan, as I've indicated before, results in a substantial reduction in this paw luminescent ratio, but when we give diazepam, there's no change in that. So we can now differentiate between the analgesia, the avoidance of the reduction of surface contact avoidance versus the sedative effects, and that is extremely useful. So one of the models that we're trying to use is to apply to our mice exactly the same kind of test that we do to humans. And to do that, we inject capsaicin into one hind paw. Capsaicin is the pungent ingredient in chili peppers, and it activates TRPV1, which is one of the transducer channels that are present on nociceptors. That's why chilies cause a burning sensation, because they activate nociceptors. And we apply this at a very low dose. This is a fraction of the dose that almost every other study does. And this is exactly the same dose that we would give in a phase 1 study to human volunteers. And when we do that, we get a substantial reduction in our paw luminescent ratio, so that the paw that has had the capsaicin injection, the animal avoids surface contact, and we can then test compounds to see whether they reverse that. And indeed, the compounds that I showed earlier, we can now test them, and we can actually see that some of them are very effective in preventing this reduction in the paw luminescent ratio as a measure of analgesic efficacy. And here I'd like to illustrate exactly the kind of measurements we can make in our mice. We have a naive mouse, and we have a mouse with intraplantar capsaicin, and you can, on the one side, you can see the signal is much reduced because the animal is avoiding surface contact. And the animal is doing this in a voluntary fashion. We are not forcing the animal to do anything. It is revealing to us that it has decided it does not want that surface contact. We can also look at other aspects. When you inject the capsaicin, not surprisingly, the animal feels pain, and in consequence, there is paw biting and paw licking, which we can also measure automatically in this device. So here's the vehicle control, where you can see on the paw that has been injected, the animal is biting and licking it. And as I've indicated, this is the concentration that has been given to humans, so we're not doing anything that we wouldn't do to ourselves. And then we give an analgesic drug over there, and you can see that the amount of paw licking is very substantially reduced. So we have multiple measures that we can identify the presence of pain and the reduction of pain by potential analgesics. In addition, we can pick up other aspects of the changes in behavior of the animals. So if we have a naive animal exploring by itself, we can pick up exactly where it moves and what it does, and you can see the animal, the pattern of movement of this animal. And then we've taken two drugs that are used in patients now, amitriptyline and morphine. Amitriptyline has multiple actions, including a non-selective sodium channel blocking activity, but it is used in patients, and obviously morphine as well. And in the case of amitriptyline, the animal is heavily sedated. So this is at the exposure level that is given to patients, and the animal hardly moves at all. Whereas when we give the standard morphine analgesic dose, we get hyperlocomotion, which is extraordinary, that the analgesic dose of morphine is causing, in addition, major adverse effects that we can pick up automatically in this fashion. So we think in parallel to our efficacy measures, we can pick up side effects, and that is just as important, obviously, because we're looking for analgesics that have efficacy with minimal adverse effect profiles. We also have the capacity now not only to measure the behavior of the animals, but we can record from genetically targeted neurons to ensure that the compounds that we're identifying do reduce the input to the central nervous system. So we've developed what we call a spinal window, whereby we can expose the dorsal horn of the spinal cord and can optogenetically record the activity of dorsal horn neurons. We can target particular neurons that project to the brain. So we are recording the activity of those dorsal horn neurons that transmit information to the brainstem and to the thalamus, and from there to the cortex where we experience pain. And this will enable us to test whether the compounds that reduce the activity have a selective activity on those circuits that drive the sensation of pain. And this just illustrates the recordings. We can apply a heat stimulus to the hindpaw, and we can pick up the activity of these dorsal horn neurons in this fashion. So I hope I've shown you that we have now developed a platform that enables us to identify compounds that are nociceptor silencers that can reduce the excitability selectively only of nociceptors with minimal or no activity on any other excitable cells. We think that high exposure to such compounds, to nociceptor silencers, will eliminate nociceptive pain. So this will include pain during surgery, so the surgical incision will no longer be painful and can also be used in the context of acute trauma. We also think that lower exposures that don't completely switch off the pain but reduce the activity in nociceptors will have a beneficial effect in inflammatory and neuropathic pain conditions. So we are using this platform to identify nociceptor silencers. We have some promising hits and are about to embark on our medicinal chemistry phase to identify potential clinical candidates that we can then move on to clinical studies. So I'd like to end by just thanking the contributors to this platform. I have a fairly large group of investigators in my lab. Jae-Hoon Shim, who has developed the thallium flux, Selwyn Jakar, who has identified the multiple targets and also developed the counter screens, Vessi Petrova, who has been looking at our CIPN assays, Lee Barrett, who is my lab manager, who runs all our screens, David Robeson, who developed the device, Alex Zhang, who developed the machine learning platform to interpret all the data from our palm reader device, Bruno Turner, who conducts all our in vivo analysis, Rasheen Powell, who does our patch clamping, Omar Barkai, who also contributes to our machine learning approach, and David Yarmolinsky, who has developed our spinal window. This is a platform that has been developed by a DARPA-sponsored project, and part of that consortium is Harvard Medical School, Jen Smith, who runs the screening core at Harvard Medical School, Mark Natchak, who is our medicinal chemist, who used to be the head of research at Vertex and now runs the translational activities at Harvard Medical School, Corey Marr, who's helping him on the medicinal chemistry, Peter Sorger, who runs the lab of systems pharmacology at Harvard and a key member of our team, and Bruce Bean, who's our key electrophysiologist. And finally, Ilyas Singek, who is from NCATS, who has played the major role in the development of the differentiation protocol that enables us to turn pluripotent stem cells into human nociceptors, which enable us to now study pain in a dish as part of our platform for the development of novel analgesics. Thank you very much.
So thank you very much indeed. A very impressive lecture and fantastic use of technology. And it's good to see the use of phenotypic screening as well. So any questions? We have time for a few questions. Please, Urs.
Geneva. One question. Did you identify a new target with this screen besides TRPV1, substance P, more opiate receptor, sodium channels, and so forth.
Yeah, so we've identified both known targets as well as novel targets as well. And I apologize for not sharing with them because they're not published yet.
It's the same problem as with the compound X.
Yeah, exactly.
Okay.
But we are an academic group, so we will be publishing them in due course.
So, I have never heard of thallium assay. It's a fairly big ion. Does it go through sodium, calcium, and TRP channels?
So basically, you put a reporter that when thallium binds to it, you get a fluorescent signal. Okay. And so we use the thallium as a means of going through the channels, and once it's into the cell, we can then get a fluorescent signal that reports the...
It's like barium.
Yeah.
Yeah, okay.
There's a question at microphone 3.
Thank you. It's a very cool platform. With the body language AI machine learning, do you have the capability to look at facial expressions at all, like nose bulge or facial expressions of pain?
We certainly are extremely interested. Facial grimace is one of the features of pain. To see a mouse grimace, actually, you require a very intense stimulus, much higher than we prefer to do, but it is something that you can pick up. The original apparatus that David Roberson devised was bottom-up only, but we now are devising an approach that we can have cameras both from the top and the bottom that enable us to see the entire animal, including the facial changes in facial grimace.
It's very exciting.
And pick it up automatically again.
There's another question at microphone 3.
Great talk. So I have a question, but I expect you're going to show it, but I didn't see that. Is that for your in vitro and in vivo assays, the platforms, what do opioids do?
So we've used morphine as our positive control.
Yeah.
And we find that it is poorly analgesic, surprisingly. We do see some signal, but whenever we do see a signal, we see massive side effects. Really substantial alterations in the spontaneous behavior of the animal, independent of the pain-related behaviors. So morphine is exactly the kind of compound we don't want. We were using it as a positive control, and we're surprised at how relatively weak it is as an analgesic, at least on this assay, and how profoundly, what profound effects it has on the general behavior of the animal.
Thank you.
Any further questions or comments? In which case, Professor Woolf, thank you very, very much again, and please join me in a round of applause, and we're finishing right on time.